So lastly, stage 3 is cell response. So the cell's response to an extracellular signal is called the output response. Ultimately, a signal transduction pathway leads to regulation of one or more cellular activities. And the response can happen inside the nucleus or in the cytoplasm. It just depends on what the message is and what pathways it starts going. So many signaling pathways regulate the synthesis of enzymes or other proteins. So whether or not a certain enzyme gets made, that's kind of the easiest way to regulate something is let's not waste our even energy to make these enzymes if we don't need it anymore. So many signaling pathways regulate the synthesis of enzymes or other proteins, usually by turning genes on or off in the nucleus. So that final activated molecule in a signaling pathway can work as, again, what's called a transcription factor, which is what helps or stops a gene from being transcribed and then made into a protein. So here again is an example of a growth factor binding here to this receptor. So in this example, this is a growth factor. There's a whole, cascade of events, the signal reception, signal transduction, and in this case, the cell response would be activating a transcription factor. And then this transcription factor is going to transcribe this gene into a recipe card. That's what I like to, compare it to. A recipe card of mRNA, and now this recipe can get made into a protein. So that's an example of a cell response is a transcription factor getting activated and then telling the cell to make this protein, which is an enzyme, and whatnot. So other pathways might regulate just the activity of proteins rather than their synthesis. So maybe it's a shorter term regulation. It's just hey, tell this protein to go on break instead of just not even calling it in, if that makes sense. So yeah, for example, a signal could cause opening or closing of an ion channel in the plasma membrane or a change in the activity of a metabolic enzyme. So here is the big scale again with this overview and example with epinephrine or adrenaline. So stage 1, signal reception, epinephrine binds to this GPCR. And just from this one molecule getting bound, this whole series of events can happen, and it scales up this response. So the signal is transduced into different forms, usually one protein activating a different protein, and then that pulls in a second messenger, and then that activates a protein kinase, and then there's a whole cascade. And then eventually the cell response in this example was to tell the cells in the muscle of the legs, hey, we need energy now so that we can run. And so ultimately glycogen that stores a bunch of molecules of glucose gets broken down, and now there is 10 to the 8 molecules of glucose for this cell to use and run for its life. So yeah, that's overall the pathway. So signal receptors, relay molecules, and second messengers participate in a variety of pathways, leading to different kinds of responses in the nucleus or in the cytoplasm, and that is including cell division. So some cell has to get a message of hey, we need some more cells over here, you need to divide. And so that is going to lead us into our next lecture about the cell division. And so a response to a signal may not simply be on or off, even though that's kind of the easiest way for us to think of something. But sometimes it's not that simple. Maybe there's other, it's regulation that this signal is after. So there are 4 aspects of signal regulation. It's amplification of the signal, and thus the response. So if you look at this example, this one molecule, 3 different G proteins activate each this many of this, and then those each go off and make this many of this. So you're amplifying the signal. It's not just like one activating one, it's kind of like broadening out how many it makes at each of these stages. It also depends on the specificity of the response, the overall efficiency of the response, and then the termination of the signal. So speaking of like the amplification, so enzyme cascades, kind of like I showed, like just this enzyme is going to go activate these 2 different things, and then those 2 different things are going to go activate 4 different things. So enzyme cascades amplify the cell's response to the signal. At each step, the number of activated products can be much greater than in the step before, the preceding step. And different kinds of cells have different collections of proteins on their surface and inside, right? And so this allows different cells to detect and respond to different signals. So your neurons are going to respond to different things than like your liver cells or your stomach cells or whatever. And so the same signal can have different effects in different cells with different proteins and pathways. And then pathways branch and like crosstalk with each other to help coordinate incoming signals. So it's super complex, which is super interesting, and it's for a good reason. Yeah. And so this is what's meant by like the specificity of cell signaling too. So like here are examples to to mostly the same signaling molecule, but you might have different things inside the cell. So there might be different relay molecules inside that lead to response 1 in a different cell that has different proteins on the inside. It might lead to different responses in this cell. So 2 different responses in this cell because this pathway branches. And so it leads to response 2 and 3. Or in this third cell, cell C, having 2 different signaling molecules actually might like kind of cancel out or enhance a response. So maybe like the same signaling molecule as the other examples, but now there's this green one, can either activate or inhibit this enzyme and lead to a totally different response. And then in cell D, there's a totally different pathway leading to different response. So that's how, different things can go throughout the body and have different effects on different cells based on what proteins are there. And then another thing is scaffolding proteins. So scaffolding proteins are large relay proteins. So again, kind of like relay, they relay the message. These are large proteins that relay stuff. And so scaffolding proteins are large relay proteins to which several other relay proteins are attached. And so scaffolding proteins can increase the signal transduction efficiency by grouping together different proteins involved in the same pathway. So a scaffolding protein can kind of like scaffold different things in one place, and that's making it more efficient. And so in some cases, scaffolding proteins can also help activate some of the relay proteins. So here's an example. So a scaffolding protein really just kind of like serves to hold these 3 different protein kinases in place And then all three of them are going to get activated at the same time when this receptor gets activated. So it does make it more efficient, right? It's kind of like if you were at work somewhere and I don't know, I'm burning out right now. You had like some central station where people with similar jobs can like meet up and coordinate faster. Okay. Okay, so summary. The 3 stages of cell signaling: signal reception. Again, the target cell detects a signaling molecule that binds to a receptor protein on the cell surface. Step 2, signal transduction. Binding of the signaling molecule alters the receptor and starts a signal transduction pathway. And then step 3, the cell response. The transduced signal triggers a specific response in the target cell. So this is basically the same slide as at the beginning, but now hopefully like it makes more sense and you have all this other stuff to kind of fit these big steps. And so lastly also is termination of the signal. So you did this whole pathway, but you don't want it to keep doing this cell response forever, probably. So there are ways to inactivate these signals and these cell responses. So inactivation mechanisms are an essential aspect of cell signaling. If the concentration of whatever like the external signaling molecule is, if the concentration falls, then fewer receptors will be bound, and then it kind of just like shuts off stuff downstream. And then unbound receptors will revert back to their inactive state. And an example of communication and a cell response is apoptosis. So for example, cells that are infected, damaged, or at the end of their functional lives often undergo what's called programmed cell death, which is apoptosis. So apoptosis is the best understood type. Components of the cell end up getting like chopped up and packaged into vesicles that are digested by other scavenger cells. And this is on purpose because apoptosis helps prevent enzymes from leaking out of a dying cell and damaging neighboring cells. So if something's wrong with a certain cell, like if it's damaged, infected, or at the end of its functional life, then it'll get some signal like, hey, it's time for you to go, but before you go, clean out your office. And we can upcycle all that for other people's offices, whatever. I don't know why work is the best example for all this. So an example in an organism is this tiny soil worm called Caenorhabditis elegans, or C. elegans for short. And this is actually the model organism that I worked with in my thesis. Not this process, but just that organism. So in these worms, again, they're really tiny, microscopic. It looks like if you were to see it from far, it would look just like a little tiny fiber. That's how small they are. So in worms and other organisms, apoptosis is triggered by signals that activate a whole cascade of suicide proteins in the cell destined to die. So in this nematode, C. elegans, a protein called CED9. This protein is in the outer mitochondrial membrane. This serves as like the master regulator of apoptosis. So CED9 acts like a brake in the absence of a signal promoting apoptosis. So it's kind of like this protein's like in charge of apoptosis. And usually it's just like, okay, I don't need to do my job yet. I don't need to do my job yet. But then once the signal comes in, CED9 gets activated and it's like, all right, time to blow this place. Yeah. So CED9 is like typically like a brake in the absence of a signal promoting apoptosis. So when the death signal is received, an apoptosis inhibiting protein is inactivated, CED9 is inactivated, this disables the brake, and now it's like, yeah, let's go, let's get ready. And so this apoptotic pathway will activate proteins, enzymes called proteases, and nucleases. So these cut up proteins and then cut up DNA. Proteases cut up proteins, nucleases cut up DNA. And then the main proteases of apoptosis are what are called caspases. And so the chief caspase in the nematode is called CED3. So here is a visual example of this example. So this is that receptor that's kind of just like waiting and waiting. And so, oh, sorry, this is the protein that's kind of just like waiting and waiting. It's the brakes. It inhibits the activity of all of this. But when the death signal comes in, this protein, CED9, is inactivated. The brakes are off, and now all of this stuff goes into play. So this gets activated, activates a bunch of other stuff, activates nucleases and other proteases, and then, the cells can start like breaking down and recycling things. I like how it says the cell forms blebs. Like that's what it's called. Okay. So in humans and other mammals, several different pathways involving like 15 caspases can carry out apoptosis. So luckily, there's a lot of regulation so that this isn't just happening willy-nilly. And so apoptosis can be triggered by signals from outside the cell or inside the cell. And then internal signals can result from irreparable DNA damage or excessive protein misfolding. So like if something's going wrong inside the cell, this triggers like a response like, you know what, this is kind of too dangerous. We can't even fix this. And we don't want this cell dividing and passing on these errors. So let's just time to hit the big red button. So internal signals will come from, major things like that, like irreparable DNA damage or excessive protein misfolding. It's kind of better to just keep that contained. And yeah, so it seems like for good reason. Apoptosis evolved pretty early in animal evolution and is essential for the development and maintenance of all animals. So for example, apoptosis is a normal part of the development of hands and feet in humans and paws in other mammals. So there were actually, yeah, here's an example. So here is a mouse paw in development. So at first, there's all this tissue in between the digits or the little fingers. And so these cells end up going through apoptosis, and so that leads to like the division and having space between digits. And then apoptosis may be involved in some diseases, for example, Parkinson's disease and Alzheimer's. And interference with apoptosis may contribute to some cancers. So yeah, again, in cells, when something goes wrong, usually you would do this to stop the spread, but interference with apoptosis may contribute to some of these cancers, like not caring the cells dividing anyway. Okay, so that is everything, lecture 9. So this is hopefully what you can do with all this information. I know it was a lot of information, but putting it into practice, again, I always recommend using these as a study guide. So practice like, okay, describe how external signals are converted to responses within cells and practice doing it in your own words. Draw it out as a figure, explain in words, whatever makes sense to you. Identify different types of signal receptors and how they function. What are the roles of phosphorylation, dephosphorylation, second messengers? How do cells respond to signal? So that is lecture 9.